Back again. This is part 2 of this 3-part sequence I've told you about. This time we're going to get into the measurement techniques that are involved in cellular electrophysiology. This is where engineering has a pretty big role, and I think we should always think about measurement when we dive into a new topic. And there's some beautiful examples here actually of some pretty fantastic engineering that has enabled cellular electrophysiology to get to the state, the modern state that we have today. Now again, this is part of this 3-part sequence, which is reflective of the 3-part triangle that makes up a good way, I think, a healthy way to think about cardiac electrophysiology generally, and in this case, cellular electrophysiology. So experimental techniques is about measurement, and that's what we're going to dive into. You can't talk much about the history of cardiac electrophysiology without talking about the legendary, historic breakthrough research carried out by Hodgkin and Huxley. These were 2 English scientists who conducted their experiments on a squid axon that was large enough to put compared to today's standards, very clumsy electrodes, but was already much more valuable than just their experiments was how they interpreted those results, how they used experiments to really develop the entire theory and background of how ion channels actually work, which was truly amazing given the very coarse measurements they had and the limited access, as I've explained before, the lack of electron microscopy, for example. But again, these were experiments carried out in the 40s and 50s. They won the Nobel Prize in 1963, very deserved. And their way of thinking about things, this formalism we call it, is what continues throughout the field in many regards. And it's the formalism that we're going to see implemented in computer models in the 3rd part of this sequence. So Hodgkin and Huxley, very important. And hopefully you've seen the derivation of their equations before. I'll cover it briefly here and point you at resources to get more details. But like I say, they really formed the foundation of what we know about. Now, to measurement. Fundamentally or schematically, we can picture the measurement as something as simple as what you see in this diagram, in which we have a cell, in this case a circular cell, not very realistic, but there's a membrane around that cell. There's an electrode that actually impales or somehow reaches the inside of that cell and measures through some kind of amplifier system the voltage relative to another electrode, which is connected typically in the bath. So that's our reference electrode is the bath. Our measurements are made intracellularly or transmembrane, with the bath being the reference potential. And this is how all measurements that we know about more or less work in terms of measuring transmembrane potential. So they're all different forms of this. Here is a diagram of the Hodgkin and Huxley experimental preparation. So here you see the squid and the very large axons that gave them access. Now, large is a few millimeters in cross-section, just to keep things under control here. So this is not truly giant, but it's certainly giant compared to a cardiac myocyte or many of the mammalian neuron cells, which again are down on the micron size scale. A cardiac cell is about 100 microns long and about 10 microns across. So compared to that, these were giants. And so they were able to slide electrodes inside a segment of that axon, and that is one of the measurement electrodes. And then the outside was the reference, left in the bath, as I just described. And the difference between those two was fed to an amplifier, and that amplifier was recorded on paper traces. I mean, things were, again, very simple. There were no computers back then to manage all this. But this is the fundamental idea of their measurement system. And they delivered signals that look something like this. So this is a, more or less real rendering. It's a schematic, of course, redrawn, but it is the membrane potential as a function of time for a neuron. And you see it's very short compared to the action potentials we just covered for the heart cells. And that, of course, as you now can already anticipate, has to do with the channels, the currents that are involved, and the fact that they're quite different in the nerve cell, in the membrane of the axon, compared to the ones in the membrane of a cardiac myocyte or cardiac cell. The Initial experiments, of course, meant to explore the behavior, meant to say how, or explore and determine how did the action potential change, for example, as a function of sodium concentration. So here you see a sequence of action potentials, again, from the nerve, in which the concentration, artificially controlled concentration outside the axon in this case, was reduced. So we see these numbers 1, 2, 3, 4, 5. These were all action potentials recorded with progressively less and less sodium. Not surprisingly, the amplitude would go down because less sodium in the extracellular space would reduce that concentration gradient and reduce that driving force and reduce the amount of current that would flow through those sodium channels. And that at least explains a good deal of this change in the shape of the action potentials. And this is typical of the kind of experiments that cellular electrophysiologists conduct, in which they alter the extracellular, or in some cases even intracellular concentrations, and then watch the changes in behavior. And that is how they explore and extract behaviors of the channels of interest. Again, this is a whole huge topic in and of itself. We have separate courses on this topic. But suffice it to say that this is the general approach for a lot of the measurements that electrophysiologists conduct on these cells. Now let's jump over to see action potentials, measured action potentials, in this case from a paper in 1993, that describe some of the different action potentials in the heart cells, again under slightly different conditions. Here the pacing rate was altered, the concentrations were altered, doesn't matter the details here, but we can see that there's a range of different action potentials. There's heterogeneity also in different parts of even the ventricle walls. So some of these are from one side of the ventricle walls, the epicardial surface, that's the outer surface, the mid-myocardial, those are the M cells in the middle, and in the bottom are the endocardial or subendocardial cells. And you can see differences in action potential across that wall of just one ventricle. And there's a lot more heterogeneity we'll come back to, just within the different types of cardiac myocytes within a normal mammalian heart. And so we're going to have to use to this notion and then use the simulations we'll do as homework to try and explore some of these relationships between some of the changes we can make in the parameters of these action potentials and the resulting changes that result. Now, another type of measurement I'll mention just briefly here because it inevitably comes up. You'll see this term if you haven't already. It's called the voltage clamp. And the idea of this goes back to that equation I showed you in the previous lecture, in which we saw that if we could control the membrane voltage, and if we knew the concentration of ions, we would know the equilibrium voltage. And then if we could measure the resulting current, then we could derive, using Ohm's law, yet again, the resistance. And we could derive that resistance as a function of things like the concentrations, things like the membrane voltage, and even the change over time of those values of resistance or their inverse conductance. And this is the measurement setup that enables us to do this. Again, goes back to Hodgkin and Huxley. They carried out these experiments, developed this type of instrumentation. There's a second electrode. There's an electrode that applies the voltage, the transmembrane voltage, and then there's an electrode that measures the resulting current. Again, we won't go into details, but we can see here that there are ways to recreate the conditions I described in that equation and use it to make very useful measurements that ultimately helped us explain or describe the way conductance changes in the face of changes in time, changes in membrane voltage, and really explore the true nature of these ion channels. So voltage clamp, very important technique. We won't say a whole lot more than that about it, except to share a little bit of results with you. So at the bottom, you see the control voltage. So here you see the membrane voltage is changed. That's the control voltage. And then above it, you see two different sets of curves. And the top one is the normal response in what's called a ringer solution. Again, these were done in the squid axon. In a normal solution where we see these time traces, if you look very carefully, you can see that the response at the rapid upstroke of the voltage, the control voltage, is a rapid downstroke, which means an inward flowing current in that top tracing. So the top tracing is the current that were measured from this axon. And you can see that with different control voltages, the current has slightly different shapes. And again, you can pull this apart and analyze it. In the middle tracings, they actually applied a toxin to knock out the sodium channel. So the top set of currents is the total current. The one at the bottom is the resulting current when there's no sodium. So this is a highly toxic substance, this TTX. It comes natively from the puffer fish, which, believe it or not, is very popular in Japan. It's a delicacy in sushi, and if it's prepared right, I'm told it's very excellent. I'm not sure I'd trust anybody entirely to prepare it correctly, because if it isn't correctly prepared, it can actually cause great harm. This is a very toxic chemical. Anyway, if you apply TTX, that will reduce the sodium current, eliminate it functionally, and what's left over then is the current arising from the potassium. And you can see the curves are totally different shaped. They don't go down, they go down a little bit, but mostly rise up, and they stay up over time. It's also possible then to subtract those two and say the top one is the total current, the middle one is the current from the potassium, and that bottom traces the difference between the two. That difference is the sodium current. So that A minus B set of curves is a tracing showing the sodium current and how it changes as a function of the membrane voltage. So with this, we can then reconstruct or construct this current voltage relationship I talked about also in the previous lecture, only this case for the peak currents that are measured in this squid giant axon. And you see we get a very complicated IV curve that describes the behavior of the sodium channel. And this, again, this teasing apart of different currents and the effects of different currents is also what cellular electrophysiologists have traditionally done in order to understand the behavior of these currents. Now, these measurements have all been carried out on at least pieces of membrane or sometimes even whole cells. But we can go deeper than that, because we know ultimately this current is the integration of the current that's flowing through all of the channels that are embedded in that segment of membrane or in that cell. And there are many of them. The cells have many individual ion channels. It's also possible to measure the current through an individual ion channel, believe it or not. The trick here is to use another technique. It's called membrane patch clamping. You see it schematically here. Here there's a very small glass electrode that's brought down on the surface of the membrane. That opening of the glass microelectrode or micropipette is small enough that it typically contains just enough membrane to hold 1 to 2, maybe 3 individual ion channels. And once we grab that little piece of ion channel, it can actually be pulled back out of the membrane. The membrane is disrupted, but we then have at the tip of the electrode a small piece of membrane that contains a small number, sometimes even a single ion channel. And that's what allows us to make measurements of the currents in response to voltage steps. We use the voltage to stimulate the ion channel, to open it up, and then measure the resulting current. And the traces you see down below are, again, the voltage step is at the top here. This is the depolarizing voltage step that opens the channel. And then we see below it the individual tracings of current. And we see that these are noisy, they're very small currents, they're picoamp currents, they're tiny, tiny currents. But these are literally the openings of individual channels. And that, again, resulted in a Nobel Prize, of course, and really allowed us ultimately to confirm a lot of what Hodgkin-Huxley first formulated, but at a much finer scale. That's, of course, the beauty of this, that we're able to extract individual ion channels and study them in true isolation from any other ion channels that might be nearby. And so, again, these are the two major sort of types of techniques that make up electrophysiology. Now, I should briefly mention that it's also possible to make what are called optical measurements. It turns out that there are substances, there are dye molecules that we can bind to the membrane of a cell, and that ion sensitive or voltage sensitive or pH sensitive dye will change its state as a function of some control parameter. In this case, for example, it will change its fluorescence. It will actually fluoresce in the setting of a voltage or potential across it, or an electric field across the molecule that is the actual dye molecule. And that opens up many, many possibilities for doing non-invasive measurements. So instead of sticking electrodes in a cell, we simply bathe the cell in this dye stimulate the cell to excite the fluorescence, and then capture the fluorescence. And that fluorescence change is a direct reflection of the voltage change, in this case, that we see across that membrane. And again, whole big chapter here. This measurement technique we can explore at the cellular level, but it can also be scaled up, and we can do optical measurements of entire hearts. And even with multiple cameras, we can capture the entire surface of the heart using multiple cameras, all recording simultaneously. So this is a big deal measurement-wise, and optical measurements are used locally at our university, but also around the world. So that's a very brief picture on some of the measurement techniques. You can see some of them are done with these microelectrodes, some done with optical methods, different types of microelectrodes, lots and lots of depth here. We could examine at some future time if you're interested. Again, there are separate courses that go deeper into these techniques, but it's good to know, I think, as engineers, just how these measurements get made. So that was part 2. Come back if you're ready or whenever you're ready, and we'll get into part 3, which is going to be about taking all this information and actually creating computer simulations. And that's going to lead into the homework that we're going to do shortly. See you soon.